everyone. My name is Álvaro Guzmán. I am part of the editorial staff of NutriNews International. We are very happy to continue with our new series of technical interviews with Dr. Andrés Gómez. Dr. Gómez is a professor of microbial ecology at the University of Minnesota. Dr. Gómez, on behalf of NutriNews International, I want to welcome you and thank you for being with us.
Thank you. Thank you so much for the invitation.
Could you just give us a brief overview of your career path and about some of your current work at the university?
Sure. So I consider myself a microbial ecologist. That means my job is concerned with studying microbes in different ecosystems, whether that's the body of a human or an animal or a microbe living free in the environment. So my job focuses on studying the diversity and composition of microbes in different ecosystems and investigating how or why those microbes are important in maintaining the health or the phenotype or the physiological performance of that ecosystem. Again, whether that's an animal, a human, or let's say a river or a lake. I started this during my master's in Colombia. I did a master's on biotechnology, and that was my first experience with strictly microbial ecology. I studied at the National University of Colombia in Medellin, and there I worked with two professors, Dr. Gloria Cadavid and Dr. Claudia Moreno, on looking at bacterial diversity at different depths of a very contaminated soil or landfill in my city, Medellin, Colombia. So that was my first contact with microbial ecology. So then after I finished that experience, I went to the University of Illinois in Urbana-Champaign, and I completed a doctorate degree, also in microbial ecology, but this time focused on humans and non-human primates. So focused on host-associated microbes and how those microbes provided important clues to understand patterns of health and disease or physiological performance in the host. And after that, I went to the University of Minnesota as a postdoc. I worked with Dr. Ram Blegman. I continued working on computational biology and host-associated microbiomes. Then I had a brief stay at the Jake Preventer Institute in La Jolla, California, working with Dr. Karen Nelson, also focusing more on the effect of the microbiome in the health of humans and animals. And since 2017, at the end of 2017, I started here at the University of Minnesota, where my lab focuses on addressing some of these same problems. However, my focus now is more geared towards food animals, specifically swine, and investigating how the microbiome of pigs contributes to their physiological and health performance.
Okay, that sounds great. Quite an interesting path. Microbiome research has drawn great attention over the last couple of years, or maybe a little bit more. However, it's quite a broad concept that seems to hold great potential, but there seems to be a lot of unknowns regarding its real applicability in animal production settings. How would you define the term microbiome from a physiological standpoint and based on its relationship with animal nutrition and performance?
Well, so the microbiome is basically defined as the collection of microorganisms, not just bacteria, but also fungal cells, viruses, and microeukaryotes that colonize different surfaces in animals, humans, and the environment. So in the case of animals, you can call it the collection of those microorganisms living in different anatomical parts of that animal, including the gastrointestinal system, the vaginal canal, the skin, the reproductive tract, the oral cavity, the nose. So practically all of our bodies are colonized by microbes. And the recent evidence collected during the past 10, 20 years, and even dating further back from that, tells us that those microbes impact the physiological landscape of the host significantly, through either metabolites that those microbes produce, different chemicals that the microbes produce upon degrading or metabolizing different dietary substrates, but also, the interaction between those microbes, the metabolites they produce, and the immune system, so that three-way communication in terms of metabolism, immunity, and the role of microbes influences many different aspects of the physiological mechanistic functioning of the host, including the way we digest our foods, how effectively we retrieve energy from these foods, how well we're able to fight diseases, and also educating the immune system, how microbes and immune cells orchestrate a very fine molecular crosstalk to influence the way your immune system works. So we believe, through the research that we've doing during the last 10, 20 years, that the way those microbes interact in our gut and the composition of those microbes in the gut or in different parts of the body influences all these physiological outcomes that I just described. Physiological performance in terms of metabolism, but also in terms of immunity and health.
Yeah, that's perfect. Very interesting. As you mentioned before, you carried out previous research on non-human primates as well as in humans regarding ecological characterization of microbial composition. How much of what you have found in this previous research can you extrapolate into swine physiology, gut environment, microbial composition?
That's a good question. So my research in non-human primates, which is still active, and which I started during my PhD, focused on investigating patterns of microbial diversity and composition across different primate species, with a particular emphasis in those species that were closest to humans, such as gorillas and chimpanzees. I specifically focused in gorillas. And so the work that I conducted there started with an idea of exploring what's in there, because we did know very little about the kind of microbes that you found in non-human primates. So it was a characterization of those microbes, which microbes can be found in the gastrointestinal tract of non-human primates, and specifically gorillas, and characterizing those microbes from a functional perspective. That means not only answering the question of who's there, what kind of microbes we can find, but also what they may be doing to impact the host, the gorilla, right? So we started with different questions. So the first question was that overall characterization. Then we also characterize or answer the question of the influence of social grouping on the gorilla gut microbiome, so investigating patterns of microbial diversity in different gorilla social groups. And we also did some comparisons with other gorilla species, such as mountain gorillas. So we compared the microbiome of lowland and mountain gorillas. And I think the idea, like the overall ultimate goal, was to study how investigating the microbiome of those non-human primates offer clues to understand our own human microbiomes, right? So from an evolutionary standpoint, what kind of information can non-human primates reveal about how the microbiome has evolved along evolutionary time scales, and why do we have the microbiome that we have as humans in the context of health and disease?
Okay, that's great.
But I think I missed the second part of the question, which is how do we extrapolate that to the working
to the pigs or
swine.
Working with swine?
Yeah, yes. So the gorilla, if you think about the gastrointestinal tract of a gorilla, especially at the distal colon level, is it's a fermentation machine, right? So they consume lots of leaves, herbs, and fiber, and their microbiomes are very effective in metabolizing that fiber to produce metabolites that are significant and very important for the energetic landscape of the gorilla. So, for instance, short-chain fatty acids or volatile fatty acids are metabolites produced by the gut microbiome in the gut, in the gastrointestinal tract, and specifically in the colon, but along all the GI system. But the colon is one primary site of production of short-chain fatty acids. So those short-chain fatty acids play an indispensable role in the energetic landscape of herbivorous mammals. So mammal that depends on fiber heavily for subsistence, like a gorilla or a cow, can retrieve from 40 to 70% on their daily energetic requirements just from the fermentation of fiber and the production of short-chain fatty acids by the microbiome. So that was a very interesting question that we had. What kind of microbes we found in the gorilla microbiome that were very effective at processing that fiber and producing those compounds, which not only are effective in contributing to the energetic landscape of the animal, but also they're signaling molecules that can regulate many different processes, from immune homeostasis to even behavior. So I think the way we extrapolate, what happens in the gorilla with what happens in the pig is that if we think about it, depending on pigs' diets, pigs also rely heavily on fiber, for performance. So I think a way we can see, or we can parallelize the two processes is, what kind of microbes we find in the gorilla that accomplish the same function or a similar function in pigs. And we find very similar groups of microbes like Ruminococcus, Fibrobacter, Prevotella specifically. So those are bugs that are very good at processing and metabolizing fiber and producing those metabolites that I just mentioned that have a very important influence in physiological performance in the animal. So if we can study those microbes, investigate how they process the fiber and how they metabolize different dietary substrates, we probably can make pigs more efficient into metabolizing fiber and maybe producing metabolites that are more conducive to being better performers, grow faster, be more efficient with feed, etc.
That's great. In regards to your research with pigs, a great part of it has focused on the early stages of the piglet's life. Why is that so? From what you have found in your studies, are there greater limitations in regards to being able to modulate the microbial composition in later stages of the pig's life, or why is it that you have focused on these early stages?
Yeah, so the early environment, the early gut environment is plastic. It's ready to be colonized and modified by different stimuli. So the first microbes that we get as mammals, including pigs, is from the vaginal canal, the skin, and the maternal environment. Same for humans. We get our first microbes from the different microbial communities that live in our mother's vaginal canals, but also in the skin and whatever microbes are found in the delivery environment in which we're born. So the gut is born sterile, even though there's a little bit of controversy, recently in terms of the existence of the pre-seeding of the microbiome in utero. That's still under a lot of debate. But we believe, for the most part, evidence says that we may born sterile, and the first colonization of microbes happen during that delivery mode. So that very first gut environment is very plastic. It's not supposed to have other microbes there already. So it's relatively easy for first microbes to take foothold in the system and colonize them. So that plasticity of the early gut opens a lot of opportunities for manipulation of the microbiome, because later in life, once the microbiome is more established, which we believe in mammals and humans may happen around close to 3 years old, once the microbiome is more established and there is a more complex group of microbial communities, it's difficult for new members of other microbial groups to colonize and to take foothold in the system. There's a phenomenon called colonization resistance, which means that whatever new microbe whatever new influence of whatever microbial exposure we see from the environment will have to compete with the microbes that are already there to take foothold in the system. So the more plastic the microbiome, the more, let's say, simple in a way, it would create more opportunities for colonization and modification through introduction of new microbes or modulation of the microbiome by diet. Let me give you an example. One of the microbes that we see peaking in the early stages of development in pigs is lactobacillus. And lactobacillus is highly stimulated by maternal milk. And we know also that lactobacillus may be associated with probiotic properties in the gut in terms of immunomodulation and effective metabolism of foods. So it's relatively easy to introduce or to expand the levels of a very beneficial microbes such as lactobacillus during the lactation processes, because that's the main substrate lactobacillus uses for growth. But later, in later stages of development, especially after nursery and close to finishing the pig, when the microbial communities are more established, is relatively harder to either introduce new microbes or change the microbiome substantially, given the presence of microbes that are already there, colonizing and owning the system. But still, manipulation of microbiomes through dietary intervention, especially drastic changes in diets, is possible. But the effects of the early manipulation, and this is something we're studying in our lab right now, may have effects that are long-lasting.
Okay, that sounds good. And following a little bit this train of thought, you already mentioned a little bit of it, but how significant is maternal influence on this microbial composition in piglets? And to what extent is this vertical transmission determinant of the enterotypes found in piglets? And how detrimental can excessively sterile environments be during this initial colonization? Are there limits regarding the use of antimicrobials and disinfectants? And do you think these parameters could change in current production conditions, where we know that pig facilities have high biosecurity standards? Do you think there is a way to change this through the research that you have seen, or is the industry not very willing to do so?
Yeah, so my research on the early scene of the microbiome in the maternal environment focuses on those first microbes the pig acquires, during those first days of life. So we have data showing, not only us, but there's other researchers showing a similar phenomenon. We show that the way we are exposed, or the way pigs are exposed to microbes at the early stages of life, may have a very significant effect on which microbes get to colonize that first immature microbial environment. So one of our papers, and this is something that we're still following up, shows how, for instance, disinfection is a common practice in swine production facilities. Very important practice from a biosecurity standpoint. We need to disinfect to make sure the environment is devoid of potential pathogens that can take foothold in the system and trigger an outbreak of disease, whether those are bacteria or viruses. So disinfection is effective on that. But we also found that disinfection, or lack of thereof, can have a significant influence on how the microbiome is colonized at the various stages of life. So for instance, devoiding an environment from disinfection in the farrowing facility facilitates more colonization by many different types of microbes in the gut, but also in the nasal cavity. While disinfection, using common methods that are common in the swine industry, may limit the rate at which the piglet acquires new microbial seeding sources from the environment. Now, this is interesting because a similar phenomenon has been found in pigs that are raised outdoors, in contact with soil, right? So pigs that are raised in soil have a different microbiome compared to pigs that are raised conventionally, and pigs that are raised in soil may have faster maturation of the microbiome in terms of the first microbes that you see colonizing the gut. So for instance, you don't expect to see microbes that effectively digest or metabolize fiber until after the start of the nursery stages, or after weaning, when the animals start receiving solid food. Then you can see a bloom of the ruminococcus, prevotellus, and all those bugs that are very effective at fiber degradation. So what we see in pigs that are raised in soil is that the colonization of the microbiome, but those microbes, is faster. So we see faster along the life course those microbes that can be very effective from a metabolic standpoint in terms of processing different fiber stocks in the gut. So we believe the more microbial exposure can accelerate maturation of the microbiome. Now, what is the consequence of that accelerated maturation? So from an energetic standpoint, it might mean that the pigs may harbor bugs that are more effective at processing fiber earlier in life. But there's also something that for me is a little bit more interesting, which is the way our microbiome educates our immune system. So when we're born, the main thing that needs to happen so the microbes can colonize our gut and our different anatomical surfaces is that the immune system is blunted. That is the production of pro-inflammatory cytokines and highly inflammatory or pro-inflammatory immune cells is repressed. So that repression allows the successful colonization of microbes in the gut, for instance. So I'm very interested in investigating how increasing more microbial exposures at early stages of development would increase maturation of the gut microbiome faster in life, but in terms of educating the immune system. I like to see if that early introduction of more microbes can educate the immune system in a way later in life that immune system is more effective and resilient in fighting potential environmental insults from pathogens. So this is something that we're following up right now. Very similar to what happens in humans in the so-called hygiene hypothesis. And I don't know, Albert, if you heard about the hygiene hypothesis, which is being reformulated to be called the good friends hypothesis, which is basically the phenomenon by which humans in westernized or industrialized societies have become so sanitized, and by the use of disinfection and a very limited number of microbial exposures early in life, to the point that deprivation of early microbial sources or exposures is associated with the emergence of immune or autoimmune disorders later in life. So I want to investigate if more exposure to microbes educates the immune system in the early gut in the pig in a way that immune system triggers processes related to immunological memory and is able to effectively fight and recognize potential pathogens along the life course. So similar phenomenon, we believe that in humans, the deprivation of microbial exposures and the increased use of sanitation and disinfectants have impaired that molecular crosstalk that happens between our microbiome and our gut-associated immune system. In a way, there's an impairment in the way our microbes are able to recognize self from non-self, initiate or mount an effective immune response. So this is the extent of things that we're looking at with pigs by manipulating that early environment, allowing or depriving more microbial colonization, and analyzing if later in life, those microbes that were able to be exposed to more microbes are more effective in, for instance, outbreaks of Salmonella or E. coli or pairs of different viruses. This is what we're doing right now.
That's very interesting. Quite a nice topic. In regards to this that you spoke about in non-human primates, where you also looked at maybe microbial composition and its relationship with social interactions. Have you seen something like this in pigs, or like some relationship between certain groups of pigs that are living together if they share certain enterotypes? What's your opinion on this, or can this be determinant in that microbial composition, or do you think the early life transmission is much more important than any of these interactions?
Well, that's a good question. I would say early life seeding is the primary signal that your microbiome gets to start developing and evolving. But later in life, there are other stimuli. Food might be the most important stimuli. But as you say, interactions with the environment may also be very important. Interactions with other pigs, especially in different physical enclosures, how frequent is that contact with other animals. Those are things that can certainly influence the way microbes between the gut and the environment are exchanged. But I would argue that the influence of those factors is of less significance when we talk about whole microbial communities than the signal that you get from early seeding, from the vaginal canal, from feces, and from the environment, or the stimulation that you get from the diet. Sure, pathogens are transmitted that way, right? Pathogens are transmitted pig to pig or from the environment. But in terms of microbial communities, and let's say the commensals that you normally have inhabited in your gut, at least in the case of pigs, I would say that there's a stronger influence of that early seeding process through vaginal microbes, feces, or the environment, and also through diet than by social exchange or exchange between different, groups of pigs within the same pen or the same enclosure. So in order of importance, I would say that early seeding is first, then it's the diet, whether that's lactation or nursery diets, and then are those interactions or that exchange of different microbes that you have with other individuals in the same place that you live.
Okay. Coming
And you
back
also
on the
asked
point of
about
the diet.
enterotypes.
Yeah.
Right?
Yeah.
Sorry, let me elaborate a little bit more on enterotypes, because I think that's an interesting phenomenon today. So let's say about 15 or 10 years ago, I don't remember the exact date, there was a paper that came out with humans. And these researchers found that humans, regardless of diet, age, geography, are sorted into configuration of the microbiome called enterotypes. So those enterotypes are basically a structure of a microbiome that is characterized by high abundance of one, two, or three specific microbes. So the enterotypes in humans, as far as I can remember, they will characterize as some people having greater abundances of Prevotella, other people having greater abundances of Ruminococcus, and other people having greater abundances of other bugs. And that's what the enterotype, or that's how the enterotype idea came to be. Recently, the same enterotype idea has been explored with pigs. So we have seen how pigs, even within the same facility, the same farm, randomly assort into different enterotypes. And that assortment has nothing to do with the diet, because all pigs are under the same diet, with geography or environmental exposures, because all pigs are under the same physical facility, or other influences. So that's very interesting, seeing how pigs randomly assort into enterotypes or microbial community compositions that are specifically and highly dominated by one group or two groups of bacteria. We don't know, however, whether assortment of enterotypes in pigs, and we've also seen that in our own research, both during lactation and nursery stages. And I think that's the most important question to address is, we don't know whether those enterotypes have any influence in the physiological landscape of the host. So that would be a great contribution if we elucidate, or if we investigate or discover that pigs with specific enterotypes are better at performing in terms of, metabolism or immunity than other pigs. Because if that's the case, we can identify those pigs that are classified into enterotypes that are less favorable, and we might be able to envision or visualize or plan environmental or dietary manipulation strategies to move pigs from one enterotype to another. But we're not there yet, because we don't even know if those enterotypes are important from a physiological standpoint. It might be the case that they're not. That it's like in humans, you and I have very different microbiomes, and I presume you and I are healthy people. So even though we have different microbiomes, the outcome as far as physiological performance is the same. So if that's the case in pigs, then the idea of enterotype is not very valuable. But if in fact the enterotypes randomly have an influence on physiological performance in terms of health and the way pigs metabolize feeds, then I may be a game changer in the swine industry.
Yeah, that would definitely be a huge breakthrough performance-wise. In terms of modulating microbial composition, you spoke a lot about the diet being quite determinant. In terms of other nutritional strategies, for example, feed additives like prebiotics or other existing additives, how influential are these? Do you think there are new products coming out that could be game changers, or do you still believe that the diet has a much greater weight in terms of modulating microbial composition?
Yeah. So right now in the industry, there's a myriad of products that claim to influence the microbiome and gut health, which is another very vague term. Gut health can mean microbial composition, digestive, effective digestion and absorption, function of the immune system. So all of these products, whether there's probiotics, prebiotics, or the emerging category of postbiotics, which are basically metabolized or chemicals that come from microbes upon metabolizing different substrates. For instance, short-chain fatty acids are sold as postbiotics. So there's a big there's a lot of enthusiasm in the industry as far as how these additives are able to modulate whether it's the microbiome or the immune system or effective digestion and absorption in the umbrella of gut health, are able to modulate those factors to improve piglet performance. So my opinion on that is that, look, there's many of those products. Some people claim those products have been very effective, and some farmers do use many of those products on a regular basis, a combination of pro and postbiotics. And I believe to an extent, there's a lot of research backing up those claims. Those products do help, but there's instances in which those products don't work. So I think a good exercise would be thinking about the extent to which those specific additives, pre, pro, and postbiotics, can be tailored to the needs of individual animals or individual farms. I think that's the future of this feed additives in the swine industry, and also in human health. You're familiar with what we call today personalized nutrition or precision nutrition. So exactly. So I don't think pigs are different. We tend to have very anthropocentric views of how pigs work, and we believe that humans are unique in many senses. But I don't think there's not. I mean, we're not that different, physiologically speaking, from how a pig works. And we know that different pigs have different microbiomes, right? So I think the future of that industry would be, in order to accelerate the effectiveness or evaluate the effectiveness of those products, is to analyze the extent to which these interventions need to be thought of in terms of individual farms first, and maybe in a distant far future, based on the needs of individual pens or animals within a pen, right? But however, in terms of costs and yeah, costs in general, that's an impossible thing to think of right now. So we tend to formulate these diets and these additives through, whole farms or whole herds. So yeah, there's a lot of research, as I said, with very mixed results demonstrating the effectiveness of those products. But for me, I think that it's very difficult to formulate a single microbe solution to any problem. For me, it's a combination of everything. So for me, I think the future is not to investigate the function of one or two single microbes, but the function of consortia of different microbes, a handful of microbes, I don't know, 5, 10 microbes that can act or function in an integrated way, in communities, and not only the microbes, but also the metabolites they produce. So the future of these kind of products is probably something that would have the three things simultaneously, a prebiotic, a probiotics, live microbes, but also postbiotics, and there's a handful of companies that are now thinking about taking that further, that extra step, formulating diets and additives that include microbes, but also include substances that can stimulate the growth of those beneficial microbes or prebiotics, but also include those products of metabolites like short chain fatty acids and other substances or enzymes that would facilitate digestion and energy harvest by the animal um, in any um, production setting. I think that will be the future of the field. But I do believe that diet in itself, the diet is the main, is the core of the sources that can manipulate and shape the microbiome. What comes after that, those are additives that may help in those productive or health goals that any given producer has.
Okay, that's great. I think you touched a little bit on the last things I wanted to ask you. From your research, do you anticipate the availability of tools that allow real-time sampling to determine microbial profiles through the presence of certain fecal metabolites, for example? Do you think these genomic and metabolomic tools still hold prohibitive costs in terms of extending them or applying them on a commercial basis? Do you think this could change if the industry changes its view on this and if it thinks this is profitable?
Yeah, so the microbiome in swine production and animal production systems is very hype right now. There's lots of interest in different industry partners, and obviously, positions like my position at the University of Minnesota is funded with the premise of how we can manipulate or investigate those microbes to improve the efficiency of animal production systems. But I think companies are more cognizant of the importance of investigating the microbiome, even though we still don't have a clear idea of what is the translational applicability of that information. So right now, what we're doing is profiling microbiomes, in different populations, and analyzing how different stimuli, whether they're from diet, environment, or additives, manipulate that microbiome to influence physiological performance. So that is the extent of things that we're doing right now with the microbiome. But there seems to be still a lot of uncertainty as far as what is the translational importance of this. I would argue that the way companies can evaluate the mode of action of their additives is by looking at the microbiome first and see if the microbiome is a way through which their additive exerts their purported benefits. So that is one motivation for companies and producers of these additives to look at microbiomes and see, how their product can act through manipulation or modulation of the microbiome. So as companies become more interested in these kind of techniques to improve the efficiency of their standard operating procedures, there's also a need for those companies to develop their own technologies and facilities and teams to analyze those microbiomes. So, and also from producers as well, right? So companies work hand in hand with producers. So there's a couple of techniques right now that give you a very good perspective of what kind of microbes you can find in your sample, right? So those techniques have been decreasing in cost substantially during the past 10 years. You know, in the past, looking at a microbial survey of a sample could cost from, I don't know, from $200 to $500 on one sample. Right now we're at $10, $20. So that's a significant and exponential reduction in price. But still, you know, when you have a large number of samples, it can become a little bit challenging for some companies. So, you know, there's a lot of techniques that people are developing right now on facilitating the profiling of those microbes using like real-time approaches. So there's portable techniques that you can take to your facility, to your farm, and in real time profile those microbes that are there. They're still submitted to error. So there's a lot of error, a significant amount of error into in the patterns that you can get, in the sequence patterns that you can get. But there's a lot of bioinformatic techniques that allow you to get rid of that error, get rid of that error, and get in a more confident view of what's in the system. So as these, let's say, portable techniques become more affordable and more widely known and less prone to error, I would believe both companies but also producers will be exploring those methods, you know, in more detail. They're still a little bit limited as far as throughput, you know, as far as how much information you can get. But I think as a preliminary assessment in what's in your sample, they're a very good indication of that. So I think the future of this microbiome industry, or the microbiome field in the swine and the animal industry, I can foresee different companies and different even producers, you know, even companies involved in the production of animal goods. I can see each of those companies and farms and facilities having their own microbiome station within their, you know, own headquarters or facilities, so they can themselves sequence those microbiomes, characterize them, and based on that microbial composition, increase the efficiency of the standard operating procedures of additives or specific dyes that they're formulate or that they advertise or they claim to have specific effects on the physiological landscape of the animal. So I think that may be coming in midterm future, I guess.
Okay. Well, that sounds great. That's been quite a interesting review of where microbiome research stands right now and the standpoint from the industry as well. So I want to thank you for your time, Dr. Gomez. It's been quite an interesting chat. We hope to have you back for new interviews, and it will be until next time.
Thank you, Alvaro. Thank you for inviting me.